Hello, my name is Robert Hanna. I'm an associate professor at the Biodiversity Institute of Ontario and Centre for Biodiversity Genomics at the University of Guelph in Ontario, Canada. Thank you for the opportunity to present this talk on food traceability and DNA barcoding. Before we begin a discussion of barcoding, I would like to discuss the world's oldest profession, which, as this Italian fresco depicts, is of course taxonomy, the art of naming things. Early taxonomies were based on functional relationships to humans, categorizing things as edible, medicinal, or perhaps even poisonous. However, from a scientific perspective, it was Linnaeus who gave us binomial nomenclature. These genus and species names are a scientific standard for naming and describing species, which Linnaeus viewed as unmutable entities. Later, Charles Darwin gave us a mechanism, descent with modification by natural selection, to explain the pattern of diversification through time. Species exist because they've evolved. Evolution is a process. Older species tend to be more divergent and easier to tell apart. Young species have had less time to diversify, so they can be harder to tell apart. Explaining the diversity gradient we see in nature, which we seek to classify into kingdoms, phyla, classes, orders, family, genera, species, subspecies, races, even varieties. We do not know how many species exist on Earth, but over 250 years of Victorian-era efforts to describe species has resulted in a description of about 1.7 million plants and animals. However, we do not know, even to an approximate order of magnitude, how many species we share the planet with. Estimates range from 10 to as many as 100 million species. We have perhaps described only about 10% of the extant diversity on Earth. Our understanding of diversity derives from reference specimens housed in museum and herbarium collections. Traditional species descriptions are typically based on morphological differences in size, shape, and color. A result of Victorian-era classification is that the morphological keys used to identify named species are usually only applicable to intact adult male specimens. However, biological specimens come in many forms, and in the real world we need to identify not only adults, but larval and juvenile stages, as well as processed products and fragmentary remains, not amenable to analysis through traditional methods. Fortunately, new DNA-based approaches are extending our ability to identify species to fragmentary remains. This paper, Biological Identifications Through DNA Barcodes, by my colleague Paul Hébert here at the University of Guelph, set a new standard for DNA characterization of species. It can be used to identify all life stages, because there is DNA in every cell of every organism. So we can even use this tool to identify fragmentary remains or processed products. Barcoding is a method of species identification based on DNA sequencing in a short standard region of the mitochondrial cytochrome oxidase 1 gene. The hypothesis is that for that gene segment, every species will have a unique sequence or a unique assemblage of closely related sequences. This sequence is termed a DNA barcode. DNA is composed of 4 different nucleotides, and over time, different species will accumulate mutations in their genetic sequence that can be used to differentiate them. There are many uses for DNA barcodes. It is a research tool for improving our species-level taxonomy. It allows us to associate all life history stages in different sexes that may have different morphology. And it allows us to test these hypotheses about species boundaries. and identify new variants or cryptic diversity. Importantly, it is also a powerful tool for identifying regulated species such as agricultural pests, disease vectors, foods, and other invasive species. We're using it to identify environmental indicators and protected species. It uses minimal samples and can be applied to damaged specimens or even stomach contents or feces. Importantly, it is also a powerful tool for flagging potentially new species. Barcoding works by extracting DNA from a small bit of tissue, perhaps as little as a leg from a fly. The cytochrome C oxidase 1 gene is amplified using standard PCR primers and protocols. The CO1 gene is then sequenced, and we can compare that unknown sequence to a reference sequence library derived from expert-identified voucher specimens using the Barcode of Life database. Again, this procedure is amenable to any life state, including processed products or fragmentary remains. This information is stored in the Barcode of Life data system, or BOLD, an online workbench for compiling barcode reference sequence libraries that can be used to infer the identification of an unknown sample by matching its barcode sequence to an entry in the reference library. BOLD captures key elements of the barcode pipeline, including images of the reference specimen and metadata about when and where it was collected and by whom. It also captures raw sequence data and finished sequence assemblies, as well as the PCR primer sequences used to generate the barcodes. The BOLD platform also has tools to visualize the degree of genetic separation between species. We typically see little or no variation within species, but deep differences between them. Low interspecific divergences are rare, but there are examples where barcodes are shared by some closely related species, like ring-billed gulls and certain African rift-lake cichlids. But this is uncommon. Most species are old and easily identified by their barcodes. For example, humans differ from their closest ancestors by more than 5% sequence divergence. This figure depicts the combinatorial differences in the barcode sequence that makes each barcode unique. So barcodes are developing a reference library for known species that can serve as a master key that allow us to ID all life stages. We can generate identifications cheaply and quickly with relatively low residual taxonomic uncertainty. Building the reference barcode sequence library is the job of the International Barcode of Life Project, a collaboration of more than 24 nations working collectively to barcode 5 million specimens from 500,000 species by 2015. While this will not complete the library for all of life, it will allow us to identify the vast majority of species that are of socioeconomic importance. Importantly, Brazil is a flagship member of the IBOL project. The Brazilian Barcode of Life Network is headed by Claudia Oliveira at UNESP. There's a website with more information about the project. Brazil is an active member of IBOL and includes over 100 participants. And Brazil BOLD also hosts a mirror of the BOLD database. The IBOL project is housed at the Center for Biodiversity Genomics here at the University of Guelph, which serves as the secretariat for IBOL as well as hosts the core sequencing facility for the IBOL project. Here, researchers are working to reduce the cost and increase the speed and efficiency of barcoding. Today, we can generate a barcode record in as little as a few hours for a few dollars, and this price continues to collapse as we push on protocol development. We also host training programs for visiting researchers. A number of Brazilian students have been in residence here at the Canadian Centre for DNA Barcoding, and we welcome more in the future. IBOL is promoting a census of all of life and democratizing access to biodiversity information. We're also working to develop the kind of technology that would allow us to move barcoding from the lab into the field. While this picture depicting a handheld barcoder is of course fiction today, all of the technology exists to develop such a device. It simply needs to be miniaturized and integrated. And of course, the barcode reference libraries remain to be constructed. Barcoding is a powerful tool. By generating a barcode record from an unknown, we can compare it to the reference sequence library in BOLD to infer a species-level identification. Once we have a name, that name can be Googled, and we can find out all sorts of interesting information about that specimen. What species it belongs to, where it's from, what it does for a living, what its economic value might be, or whether or not it's endangered. It's the act of being able to put a name on a specimen that unlocks all of this other information about that specimen. And that is the key revolution that DNA barcoding is bringing to us today, allowing us to extend our traditional taxonomies in important ways. For example, we've used DNA barcoding to investigate species identities of seafood fillets purchased in New York and Toronto. We were able to recover a species ID for all specimens tested, but surprisingly, 25% were misladen, always with the species of a lower market value being substituted for those of a higher market value. This is a form of food fraud. Seafood fraud has been documented in Europe using barcoding, with smoked products being particularly prone to substitution. Barcoding has also revealed seafood fraud in Asia, where a number of different species were commingled under a single species name. Commingling of different crab species has been detected in Chile using DNA barcoding. And incorrect labeling is a problem in Brazil too. In this study, DNA barcoding revealed the incorrect labeling of around 80% of samples analyzed, with mislabeling being more pronounced in fillets rather than whole fish. Not surprising. DNA barcoding has demonstrated that Amazonian species are also being commingled and exploited as a single species. Ongoing studies of this nature suggest the problem with seafood fraud is a global concern. Studies like this one show that it is also a problem in South Africa. But market substitution is not just an economic or ecological problem. It can also impact human health. For example, a couple who bought what they thought was headless monkfish at a market in Chicago ended up in critical condition at the hospital. The U.S. Food and Drug Administration recovered the meal remnant from their stove, but this fish soup could not be easily identified. They sent samples to us at the University of Guelph for barcoding, which were revealed to be pufferfish. This finding was reinforced by a toxicological assessment conducted by FDA, which revealed the presence of tetrodotoxin, which comes from pufferfish. Together, these findings demonstrated that it was not headless monkfish, but in fact pufferfish that had been sold in the market that had poisoned the consumers. multiple times. This has prompted the U.S. Food and Drug Administration to add DNA barcodes to their regulatory fish encyclopedia. FDA formally announced its use of DNA barcoding as a regulatory protocol on its website in the fall of 2011. The protocol has been validated for regulatory use. The FDA is now extending barcode characterization of other species, such as the Dirty 22, insects that are known to spread foodborne pathogens. Despite these advances, seafood fraud continues to be a problem here in Canada, where regulatory agencies have been slow to adopt new technology. Even high school students have used barcoding to detect food fraud. The technique is relatively simple to employ. And it's not just seafood. We're finding that food fraud is a problem in other domains as well. About 1/3 of herbal teas students tested in this paper generated DNA identifications not found on product labels. If you've ever wondered why herbal tea tastes like hay, it's because many of them contain alfalfa, but virtually none of them list that on their label, on their list of ingredients. Barcoding has also revealed that many natural health products are also substituted. This is an area that is currently unregulated, but many of the items that have been substituted either have no medicinal value or in some cases can even be toxic. The community is increasingly recognizing barcoding as a new tool for food traceability. And they recognize that cases of food adulteration, when reported by the media, have a great impact on public opinion. And this is prompting an interest from industry in authenticating ingredients in their products. There are many drivers of traceability in the food sector. Not the least of which involves a concern for endangered species. A Google Global Impact Award was recently given to the Smithsonian Institution in support of this Barcode of Wildlife project, which aims to increase awareness about the potential for using DNA barcoding as a wildlife forensic tool. Importantly, DNA barcoding has been used to identify sharks and rays. Many endangered species are showing up in the seafood supply chain. And genetic evidence of illegal trade in protected whales has been found linking Japan, the US, and South Korea. Barcoding is an important tool in bridging the gap between conservation genetics and law enforcement. Importantly, barcoding has been validated for use in forensic genetic species identification. Validation demonstrates that a procedure is robust, reliable, and reproducible. PCR amplification and DNA sequencing are robust methods, which produce successful results a high percentage of the time. They're also reliable methods that produce accurate results, and they are reproducible methods, producing similar results each time a sample is tested across different laboratories. The forensics community has also been an early adopter of barcoding to begin to identify things like forensically important blowflies, which can be used to determine the post-mortem interval and time of death of a corpse. The forensics community has urged academic collaboration in building the reference sequence libraries for the species of concern to the forensic community. They've also recommended the creation of centers of excellence to pursue this agenda. The Daubert standard fits well with barcoding. Barcoding enhances prior art, is easily explained to a judge, provides an independent test of morphology, and can provide a probabilistic assessment of error given reasonable sample sizes. In summary, Barcoding is developing as a standardized analytical method that is easily replicated across different labs and has many potential applications. New next-generation DNA sequencing technologies promise an enhanced ability to analyze mixtures. Through massively parallelized pyrosequencing, we can identify all of the species that contribute to a particular food product, if those foods have been barcoded and are available for lookup in a reference sequence library. So what are the next steps? Perhaps an interagency working group, a center of excellence, a regulatory community presence in Iowa. This recent publication from the Grocery Manufacturers Association of America suggests that information clearinghouse partnerships are needed to assess the supply chain, because it is not always clear where in the supply chain food fraud is occurring. Today, under global food processing regimes, many different hands touch our food products before they arrive on our plates. If you would like to know more about DNA barcoding, we would urge you to attend this upcoming conference in Kunming, China. The 5th International Barcode Life Conference only happens once every 2 years. In closing, I'd like to share this cartoon. It reminds me of the time that I've spent in various metropolitan areas wondering just exactly what's being served. With DNA barcoding, we can now finally answer that question. Thank you.
Convido a todos agora, então, a tomar sua cabeça fora e voltar para a discussão. O doutor Robert Schneider, que agora não vai participar da discussão através do Skype.